Hello, students. I hope you are doing well. This is a recording to give you a feedback on the biology exam that you took, the first semester exams. Okay, so that as you're at home, you will know at least where you went wrong and then correct them. If there's a need for further highlighting of some points you are not so clear about, then you can post it on our WhatsApp page and then we can look at it, address it. Okay, so we are going to look at paper 2 first, and then paper 1, the multiple choice aspect. So if you are ready and I'm ready, we'll look at question 1 of paper 2. The first question is: in harvesting fish to be processed and exported to another country, the following practices were carried out by the harvesters. That is, you suggest why each process is carried out. So I, starving the fish on the day of harvest. I, removing scales. I, freezing the fish. V, removing intestines. V, increasing fish feed 2 weeks before harvest. Then we look at B. Describe the process a cell uses to get rid of large waste particles or to secrete hormones that are too big to pass through a protein channel. Then C. List the major taxa used in the hierarchical classification of organisms, starting from the smallest to the largest. And then to give two differences between passive and active transport. And then D, state 3 characteristics of the cell membrane according to the fluid mosaic model. So this is for question 1. So let's look at the answers that for this part of the question or paper 2, which is question 1. So what are the answers? So the reasons for the practice is carried out during fish farming. So starving the fish on the day of harvest to empty the guts and reduce fecal waste in the fish. This, you can also say that this maintains cleanliness and reduces bacterial contamination during processing and transport. Okay. Removing the scales. This will make the fish easier to clean and process. Also removes appearance and makes the fish ready for cooking or export standards. Freezing the fish. Why do you have to do that? To preserve the fish by slowing microbial growth and enzyme activity. And this prevents spoilage and keeps the fish fresh during transportation. Okay, so that's for freezing the fish. Then removing intestines. The intestines contain many microorganisms and digestive enzymes that can quickly spoil the fish. Then removing them increases shelf life and improves hygiene. Okay. Yeah, so in most cases, this is done for the reasons above. Though some people may leave them to probably increase the size or the mass of the fish when it's being weighed or something like that. Okay, so whichever way, but this one is for removing the intestines. So increasing fish feed 2 weeks before. Okay, so to increase the body weight and size of the fish before sale, also referred to as the table size or market size. This improves quality and market value for export. Alright. Then we go to the second question, which summarizes here that process used by a cell to remove large waste particles or secrete hormones. You need to recognize that this is a process of exocytosis. So that should be somewhere in your discussion, can come first, like we have here, whatever. So during exocytosis, materials such as hormones or large waste particles are packaged into membrane-bound vesicles inside the cell. The vesicles move toward the cell membrane and fuse with it. That's referred to as docking. The contents of the vesicle are then released outside the cell. What happens to the vesicle? So this vesicle membrane becomes part of the cell membrane after fusion. Okay. Of course, this process requires energy in the form of ATP because it is an active transport mechanism. Right? So that's how you can go about it in different ways. It depends on you, but then this is the chronological order, the right order for you to talk about it. The next question, if you've not forgotten, we have it here summarized that what's the major taxa in the hierarchical classification, smallest to largest. The smallest having the smallest numbers and those having the largest numbers. So we start with species, genus, family, order, class, phylum or division, kingdom, then domain, right? By way of the hierarchical classification. So that's what you have. So you should have it in this order to score, right? Okay. Hello. I hope you are with me. All right. Okay. So if you feel like I'm jotting things down, you can do that, or you can save or download the video for later revision. You can come back to it and then look at it again. You have to state two differences between passive and active transport. So you're giving some 5 of them here so that you can enrich or update your knowledge. Then does not require energy. For passive, active transport requires energy. Passive transport moves substances from high to low concentration. Active transport moves substances from low to high concentration. Passive occurs along the concentration gradient. Active transport occurs against the concentration gradient. The next one, not so high grade, but it's okay, includes diffusion and osmosis. That's examples, not so much of a strong point because, Then for active includes, generally you can talk about bulk transport, and then they involve the pumps. You have the sodium potassium pump, you have the sodium pump, the potassium pump, the voltage gated pumps, calcium pumps, and so on. So many pumps, and most of them, all of them requires energy being used in bulk transport. Alright, then may occur through channels without energy use. So we talk about the aquaporins, basically a lot of plant cells or in plants allows water to move freely along the concentration gradient. This time not through the membranes but through channels, and call it it's also part of the passive transport, even though it's through channels, so the membrane proteins. Okay. All right. Uses carrier proteins or pumps with energy. Uses carrier proteins or pumps with energy, as we have looked at earlier. And then here we are to give characteristics of the cell membrane according to the fluid mosaic model. So one, that the membrane is made up of what? A phospholipid bilayer. Okay. The phospholipids, the next one, the phospholipids can move sideways, making the membrane fluid. 3. Proteins are embedded within the phospholipid bilayer in a mosaic arrangement. So they are just stacked within the layers. Okay. Some going through, some at the periphery. That the membrane is selectively permeable, allowing only certain substances to pass through, and so on and so forth. Okay. Alright, so alright, so onwards to question 2. Alright, shall we? Okay. AI to AI. State three components of the cell membrane and a function for each. I give a function for each. Right. Then AII. What is the difference between a fry and a fingerling? What is the difference between a fry and a fingerling? Then 2BI. A learner is provided with fingerlings and mature fish to cater for. Explain in 3 reasons why these two should be kept in different aquaria or ponds. So we have a fry or a fingerling and then a mature one. Why is that during the grow out stage you should keep them in different aquaria? BII. Explain how amoeba undergoes phagocytosis. Alright, so that is for question 2. Okay, that's question 2. So why don't you look at the scheme with respect to question 2. 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 So we start from here. Right. So the components of the cell membrane, their functions. So added some more so that again you can update your knowledge and then on this one. So components of the cell, phospholipids and the function. It form the basic bilayer structure and act as a barrier between the cell and its environment. Then we have proteins. Okay, we have proteins. One of them being integral proteins. What does it do? It transports substances across the membrane through channels or carriers. Then we have peripheral proteins. Those are the surfaces of the membranes. Provide support, enzymatic activity, and cell signaling. Cholesterol maintains membrane fluidity and stability. Glycoproteins involved in cell recognition and receptor function. So as we said, we don't want to use help in our discussion, so you can just go straight to the point and just see for cell recognition and receptor functions. Okay, and glycolipids assist or involved in cell identification and membrane stability. Again, you will not use assist here. So this function, that's its function for cell identification and for cell identification and membrane stability. Okay, so that is also for that. Then go to AI, where we ask the difference between a fry and a fingerling. A fry is a young fish that's recently hatched and is still very small and delicate, while the fingerling is a more developed juvenile fish, larger than a fry, usually about the size of a finger. Alright, so that we call it fingerling, right? Okay, so BI. Reasons why fingerlings and mature fish should be kept in different aquaria or ponds. Mature fish may eat the fingerlings because they are smaller and vulnerable. We also write that fingerlings require softer feet, while mature fish require larger feet. That also means smaller feet for the fingerlings. Mature fish compete more strongly for food, causing fingerlings to starve. Then you can also write that fingerlings need different environmental conditions such as shallow water and gentler water movement. Separating them reduce stress, injuries, and disease transmissions among the and more effective record keeping. Effective and efficient record keeping. It prevents catching of smaller fishes during harvesting. Okay. Alright, so those are some of the reasons to give, I think, 2 or 3 or so. Right, then look at 2bii, how amoeba undergoes phagocytosis. So earlier on there was a question relating to exocytosis, how hormones and other materials come out of the cell. This time we are looking at how materials which are in large forms, more bulky, now comes into the cell. Okay, and these are so large they cannot go through the phospholipid bilayer of the cell membrane. So how do we get them in, since these are also important as in amoeba, using phagocytosis? So an amoeba extends the pseudopodia around the food particle. Okay, the pseudopodia surround and engulf the particle, forming a food vacuum inside the cell, or a vesicle, right? If we're referring to other cells, white blood cells, macrophages or microphages, we're talking about vesicles. But in amoeba, special vesicle here is the food vacuum. Alright. Now this food vacuum captures the food, engulfs the food, brings it in, so into the cytoplasm. Or next, digestive enzymes enter the vacuole and then digest the food. Okay. Useful nutrients, which you say the end products of digestion, are absorbed into the cytoplasm, while the undigested materials, which are still in the food vacuole, are expelled from the cell. So what that means is that the food vacuole, just like we saw in the membrane, the hormone in exocytosis, where hormones are secreted outside the cell, they also have this time waste material, so you don't want them in the cell, so they move close to the cell membrane, like we talk about exocytosis, where the membrane of the vesicle, the food vacuole, fuses with that of the cell membrane, release the waste to the outside of the cell. Okay, so this will be interstitial fluids and blood vessels and so on, mainly interstitial fluids around the cell. Okay, for them to be expelled later when they enter the blood, it carry to the excretory organs to be released. Of course, that's in humans, but in amoeba, these waste materials will just enter the water surrounding them. Okay, all right. Okay, so this ends the question 2. This ends question 2. So we're going to look at question 3. Take a look at question 3. Take a look at question 3. Okay, it begins with 3a. During the microscopic practical, a student observed air bubbles instead of cells on the slide. I explain how improper slide preparation may have caused this problem. I describe how this can be prevented. Then B. Why is it dangerous to inject a patient with a large volume of pure distilled water directly into their bloodstream? Explain using the concept of tonicity. Okay, so I will look at 3, then we'll come to 4 very soon. So quickly with 3, with the microscopic slide. How improper slide preparation causes air bubbles. Okay, so air bubbles may form if the cover slip is dropped directly onto the slide on the sample that you have, especially in wet mounts or the temporary slide, the wet mount, instead of lowering it gently at an angle. Also, excess air trapped between the slide and the cover slip appear as bubbles under the microscope and may be mistaken for cells. Okay, so that's how the improper slide preparation will cause air bubbles, trap air within the sample. Good. Then, how can we prevent this? So to prevent this, lower the cover slip slowly at an angle, about say 45 degrees, using a mounted needle or forceps. Then ensure enough mounting liquid is placed on the slide before adding the cover slip to prevent trapped air. Right, so these are some of the ways we can do that. Just carefully lowering the cover slip at an angle onto the sample that you are preparing, having enough mounting liquid. Good. Then 3B, why injecting pure distilled water into the bloodstream is dangerous? So let's look at that using tonicity to explain. So 1, pure distilled water is hypotonic compared to blood plasma. Water moves into red blood cells by osmosis because the cells have a higher solute concentration, that's hypertonic, or low water potential than the distilled water. The red blood cells swell as excess water enters them. 4, the swollen cells may burst, a process called hemolysis. Alright. So this will happen. The hemolysis reduces the blood's ability to transport oxygen and can cause serious organ damage or death. Okay, organ damage or death to the person. Okay, so that is for the I think that ends question 3, I think, and go back to have a look at it. Yes, so that's question 3 and that. Then we go to question 4 quickly. AI, another application question. The farmer applies too much fertilizer to his or her maize plant. When it hadn't rained, the plant wilted and died after 1 week. I asked to explain this occurrence. Then we had a classification of biological keys. Below is a typical NAMED key used for identifying common vertebrate groups. Okay, can also be a dichotomous key, NAMED dichotomous key. So this is what we have. 1A, body covered with hair or fur. So that's A. Then so you should identify A. What is A basically? Then 1B, body not covered with hair or fur. Go to 2. So at 2 we have 2A, 2B. 2A, body covered with feathers. We identify the organism as B. Then 2B, body not covered with feathers. Go to 3. So we jump to 3. 3A, 3B. So 3A, skin moist and smooth. Leaves both in water and on land. We have C. 3B, skin dry or covered with scales. Go to 4. Then we have 4. 4A, body covered with dry scales. Lays eggs on land. So we have D. 4B, body covered with scales. Lives only in water, but breathes with gills. You have E. Now you have to use this key to identify organisms A, B, C, D, and E. Use the following where appropriate. So you have been given some options here. You have mammalia, you have fish, you have aves, vertebrates, amphibia, insecta, reptilia. Okay. And then why is this a numbered key? IV states one limitation of using a dichotomous key. Then BI, a student writes the scientific name of lion as Panthera leo. Explain why this name is incorrect and rewrite it correctly. Differentiate between bilateral and radial symmetry. Last but not the very least, list 3 factors used in classifying organisms. Okay, so we go to the scheme, question 4. So let me just put 4. Okay, so let's look at the answer for 4AI from the scheme. When too much fertilizer was applied, the concentration of mineral salt in the soil became very high, making the soil solution hypertonic compared to the cell sap of the maize plant. So here we are talking about the root hairs. The vacuole or the cytoplasm now becomes hypotonic. As a result, water moved out of the root hair cells into the soil by osmosis instead of entering the plant. The plant cells lost water and became plasmolized, causing loss of tubal pressure without sufficient tubal pressure. Without sufficient tubal pressure, the maize plant wilted. Since it did not rain, the excess fertilizer salts were not diluted or washed away, so the plant continued losing water. After prolonged dehydration, the tissues dried out and the plant died. Okay, so this is what we're expected to write, okay, the step-by-step manner to account for all the marks there. Then 4A identification of organisms using the key. So the key given, the organisms are A, mammalia, B, apes or birds, C, amphibia, D, reptilia, then E, fish. Okay, all right. So I'll go to I. Why this is a numbered key? It is called a numbered key because the identification steps are arranged in numbered pairs, guiding the user from one step to another until the organism is identified. Alright, then AIV. Now you should give one limitation of a dichotomous key. So a dichotomous key may give incorrect identification if the organism has unusual features. Right? So the organism has some strange features, not so common, or if the observer makes a wrong choice at any step. Okay. Sometimes when you have very large number of organisms, if you have so many organisms to identify, the option of numbered key is preferred. You go the numbered key rather than dichotomous key. So the dichotomous key may not be suitable for large numbers of specimens or organisms to be identified. Now the scientific name of lion. Say the name Panthera leo is incorrect because in binomial nomenclature, the genus name begins with a capital letter, while the species name must begin with a small letter. So the correct scientific name is Panthera leo. Panthera leo. So the small letter should begin the species name. Unlike we have in the question, so you just have to change that and so on. Alright. Then B, I will look at some differences between bilateral and radial symmetry. So bilateral, the body can be divided into two equal halves in only one plane. Okay, whilst in radial symmetry, the body can be divided into equal halves through many planes. Okay, then common bilateral symmetry, common in organisms such as humans and insects, usually those organisms with the head, with the head, or such organisms where the nerves and are concentrated at the anterior part of it, and that forms a head, right? That forms a head, or that's those kinds of organisms referred to as cephalo. Okay, right. In radial symmetry, common organisms such as starfish, jellyfish. Usually in bilateral symmetry again, we have organisms that are more streamlined. Okay, they are more streamlined, whilst in radial symmetry they are not, or they are rounded, spherical, and so on. Okay, those are some of the differences between the two symmetry. Then DII, 5 factors used in classifying organisms. So we have morphological characteristics, and the body structure. 2, mode of nutrition can also be used, type of reproduction, the cell structure and organization, the genetic or evolutionary relationships can also be used. Okay. Alright, so this is the end of the subjective part. Okay, the subjective part of the test. So we now look at the multiple. Alright, so now look at the multiple choice. So we just put the scheme and then the question, just juxtapose them so that it will be easier for us to refer to the answers. So the first one, bulk transport includes say diffusion and osmosis, endocytosis and exocytosis, filtration and diffusion, the osmosis, and then active. Alright, so the answer is endocytosis and exocytosis. Okay, these are some examples of bulk transport. Then 2, facilitated diffusion involves. So we have up here ATP, B, carrier proteins, C, ribosomes, D, DNA. Okay, so we have carrier proteins. So we know that even though it's a diffusion, the substances move along the concentration gradient. They usually cannot or do not go through the phospholipid bilayers or the membranes. They go through proteins, like the carrier proteins, still along the concentration gradient without the use of energy or ATP. As we said earlier, example is like the aquaporin, which allows water to go through, very common in plants or plant cells or plants generally. Alright, next one. Which process moves large particles into the cell? Okay, large into the cell. So the diffusion, osmosis, endocytosis, exocytosis. Endo means inside, so that's our choice. That should be your choice. Okay, then the next one, number 4. Which practice And the nursery stage involves choosing fish with desirable traits like fast growth and disease resistance to produce superior offspring. With this you are choosing. So definitely it has to be what? Selective breeding. Okay, selective breeding. So that is B. Amoeba reproduces by budding, binary fission, conjugation, fragmentation. So usually sexually it's reproduced by asexual means, and that is binary fission. That is B, option B. 6. Which condition will lead to a faster rate of diffusion? Large particle size, lower temperature, steeper concentration gradient, increased membrane thickness. So steeper concentration gradient. So we have two media with very high concentration of some molecules, very low at the other end. They are going to have really fast diffusion occurring. The rate of diffusion will be very high. Number 7. What is the primary function of a grader in a fish farm? To count harvested fish, to sort fish by size, to manage stock density, to increase dissolved oxygen levels, to prevent water seepage. So our option choice will be to sort fish by size, to manage stock density. Go to 8. Which of these is an example of an artificial habitat used in aquaculture? Mangroves. B. Coral reefs. C. Recirculating aquaculture system. RAS. Brackish water lagoons. So C will be our choice. Recirculating aquaculture system. RAS. The rest are all natural habitats. Number 9. Why is stocking density management critical during the growth out stage? Why is stocking density management critical during the growth out stage? So A. To make harvesting faster. B. To prevent overcrowding, which causes stress and diseases. C. To encourage the growth of algae. D. To reduce the need for water filtration. So our answer is to prevent overcrowding, which causes stress and diseases. Alright, we go to number 10. We go to 10. Number 10. What is the biological principle behind preserving fish using a concentrated salt solution, otherwise known as brine? It increases the fish's temperature. It hydrates the fish cells. C. It dehydrates the fish and prevents spoilage. Spoilage. I think something is missing here. It dehydrates the fish and prevents spoilage. Let's say by or through microorganisms, something like that. Through spoilage of microorganisms. I think there should be something here. Right? Microorganisms from growing. Prevents spoilage. Dehydrate the fish and prevents spoilage. Prevents spoilage. Dehydrate the fish and prevents spoilage. Just even preventing spoilage should have been okay, or would have been okay. So what they do is to stop or reduce the microbial activities and so on. So we can just just ended them, and that's fine. Let's go on. So then D, it adds nutrients, essential nutrients for consumption. So C is the right option. It dehydrates the fish and prevents spoilage, by microorganisms, or prevent microorganisms from growing on it. Okay. So that once they grow on it, they are going to release enzymes on it, and then they will digest and feed on it, causing it to rot, spoil, and so on. A solution that has a higher concentration of water than the cell it surrounds is hypertonic, isotonic, hypotonic, concentrated. So that has to be hypotonic. If it's hypotonic, it has high water potential. So water is going to move from there to the other medium or region. Right, go to 12. 12. Which part of the compound light microscope does contain a lens? A mirror, nose piece, condenser, diaphragm. Mirror, nose piece, condenser, and diaphragm. This option is not right. This option is not right. So it has to be a condenser. A condenser, right? But the nose piece, they only contain the objective lenses. Okay, let's provide that the space for the objective lenses. Okay, so it holds them, but it doesn't by itself contain a lens. It's the objective pieces or lenses that contain, of course, a lens, right? Okay, so it's a condenser. Calculate the objective lens power used to draw an amoeba of total magnification 60 times if the eyepiece is 15 times. Total magnification of 60 times, the eyepiece is 15 times. So what do you have? Times 10, times 4, times 40, times 100. So the total magnification is equal to the product of the eyepiece power or lens and the objective piece or lens. You multiply the two to get the total magnification. This time you have the total magnification and then the eyepiece. So to find the objective lens power, then you need to divide 60 by 15, right? And that gives you 4 times 4, right? So I think it's an error here as well. We have times 4, should be times 4, not times 10. Times 4, not times 10, right? Which harvesting method is best described as using large nets to capture fish during the final drainage of a pond? So we have hook and line, we have bamboo traps, we have seines, and we have aerators. So we have seines that will be used that. So we have seines, large nets, seines. In sustainable management, why is desilting a natural water body is important? Okay, why is desilting a natural water body important? Okay, let me take it again. In sustainable management, why is desilting a natural water body important? So A, to decrease primary productivity. B, to ensure easy movement of fish and sunlight penetration to the bottom. C, to reduce dissolved oxygen levels. D, to introduce more natural predators. So it's B, to ensure easy movement of fish and sunlight penetration to the bottom. Okay, to the bottom. 16. At which stage is the weaning of fry from natural feed to formulated feed typically performed? So that's the nursery stage. The nursery stage. At which stage is the weaning of fry from natural feed to formulated feed typically performed? So processing stage, nursery stage, marketing. So that's the nursery stage. Next one, number 17. Artificial classification is based on A. Evolutionary relationships, B. Many characteristics, C. Few observable traits, D. Genetic analysis. Okay, so there are few observable traits. Yes, few observable traits. 18. In a hypotonic solution, water moves. In a hypotonic solution, water moves A. Out of the cell, B. Into the cell, C. Equally, both ways, D. Nowhere. It falls into the cell, into the cell, into the cell. So when you place the cell, red blood cell, plant cell, onion, dermal cell into a hypotonic solution, then water is like distilled water, and then water is going to move into the cell. Number 19. In the dichotomous key, each statement is called a couplet or couplet, pairing, lead, division. So each statement is a lead. Each statement is a lead. Number 20. Phylogenetic classification is based on habitat, evolutionary relationships, size, feeding habits. So it has to do with what? Evolutionary relationships. So that brings us to the end of the review of the exam. Okay. Feel free, come to the recording as many times as you want, or however you want to, maybe write it down, the answers down, or whatever, so that you can have it. You just want to make sure that by the time you come back to school, we are done with this one, and then you can move on to other things. You don't want to spend too much time reviewing, the exam paper before we move on to the next semester's business. Okay, so try and really familiarize yourself with these feedback or this information. Also, feel free to get back to me if you feel probably some other answers may be suitable, so that we can look at it. Okay? Alright. So, see you in the next one. Bye.